Now I'd like to introduce my colleague, Dr. Aman Chuahan. Aman received his MD degree from Kasturba Medical College in India. He then completed a dual residency in internal medicine and pediatrics at Louisiana State University. He also completed a fellowship in hematology and oncology at the University of Kentucky, where he specialized in neuroendocrine neoplasms. Additionally, Dr. Chauhan completed a cancer therapy evaluation program, physician internship at the NCI that focused on designing clinical trials and leading clinical research projects. He is a member of the American Society of Clinical Oncology, American Association of Cancer Research, and the North American Neuroendocrine Tumor Society. Aman?
Thank you so much, Paul. Greetings from Kentucky viewers. My name is Aman Chuahan. I'm one of the medical oncologists. Neuroendocrine cancers are area of my clinical as well as research interest, and I direct our neuroendocrine tumor Theranostics program at Markey Cancer Center. Today, I would be talking about a very interesting, novel, and exciting phase 1 and randomized phase 2 clinical trial in neuroendocrine cancers. Next slide. Let me prime the audience with neuroendocrine cancers. These are actually a group of cancers which can originate anywhere in the body, from head to toe. However, pancreas, lung, and small bowel forms the top 3 sites of origin for these cancers. These are relatively rare, although the incidence of these cancers is on the rise. However, the prevalence is markedly disproportionate and is pretty massive, and I will touch on that in a bit. The treatment options are unfortunately very limited. Next slide. Here, I would like to review the SEER database review published in JAMA Oncology by my friend, Dr. Dasari and MD Anderson. What you see on the top blue line is the incidence of most common cancers in the United States over the last 4 decades. And fortunately, what you see here is that the incidence of major cancers have plateaued in last 40 years. In fact, the incidence has actually declined in last 5 to 6 years, probably because of improved lifestyle, decreased smoking, etc. However, I would like to bring your attention to the bottom line in yellow, and it is very concerning. That is the incidence of neuroendocrine cancer over the same time period. And the incidence of NETs have actually increased 6 to 7 folds in the same time period. Next slide. What is even more concerning is the cumulative prevalence. Incidence of NETs might be 8 to 10 per 100,000 at present, but since majority of these patients live for several years, the prevalence of these cancers outweigh its incidence by a mile. In fact, here in an op-ed published in JAMA Oncology by us last year, we reviewed the prevalence of various GI cancers and other solid tumors, and you would be surprised, neuroendocrine cancer prevalence is second to colorectal cancer in terms of prevalence of GI origin neoplasms, higher than gastric, pancreatic, esophageal, and hepatobiliary cancers. At present, there are over 170,000 patients who have been diagnosed with neuroendocrine cancer at some point. Next slide. Now, since we know the growing burden of this relatively rare but heavy prevalence disease, let's kind of familiarize ourselves with different subtype of neuroendocrine cancers. As I mentioned before, it's not one disease, but it's a group, it's a bouquet of disease or a spectrum of disease, which can vary from a indolent spectrum, low-grade, slow-growing neuroendocrine tumor to a very aggressive, fast-growing neuroendocrine carcinoma. So those are the two spectrum of the diseases. Neuroendocrine carcinoma, some of the prime examples you might have heard, for example, small cell lung cancer, which is about 14 to 15% of all lung cancers. It's a very aggressive disease considered as a recalcitrant cancer by NCI and high critical unmet need. Neuroendocrine tumor is another unmet medical need, and these patients, although relatively slow growing, certainly can cause fatal illness. The two prime examples are on your screen. Steve Jobs, the founder of Apple, unfortunately succumbed to pancreatic neuroendocrine tumor whereas Irfan Khan, Oscar-nominated actor who was in Life of Pi, unfortunately succumbed recently to high-grade neuroendocrine carcinoma. So these diseases certainly are fatal if not treated properly, and unfortunately, the tools are very limited. Next slide. Over the past several decades, we have made some increment improvement in terms of FDA-approved treatments. But if you compare it to any other cancers, be it common cancer or rare cancers, this is a very dismal progress. In fact, most of these drugs, for example, streptozotocin, interferon, are no longer even used in clinics today because of poor side effect profile. So there is a lot of catching up that we need to do in neuroendocrine cancers. Fortunately, with new promising drugs, the drug development is catching up and we are seeing some promising studies, but they have not really yet resulted into FDA-approved drugs. Next slide. And this is where Seneca Valley virus comes in. Now, you've heard a little bit about Seneca Valley virus, the preclinical data around it. I would just focus primarily on Seneca Valley virus and role in neuroendocrine cancers. It was initially tested in high-grade neuroendocrine cancers like small cell lung cancers. This is the immune deficient mouse model, the SCID mouse model, growing small cell lung cancer tumors. And just one injection or one infusion of Seneca Valley virus led to complete remission in these mice. These mice survived for over 200 days and had no significant toxicities. These data were presented by Dr. Charlie Rudin and Paul at various medical meetings. Next slide. Not only preclinical data, there is a considerable amount of clinical human data which supports that Seneca Valley virus might be a fantastic therapeutic intervention in neuroendocrine cancers. Precisely, there have been 2 phase 1 and 1 phase 2 study. The phase 1 studies included various types of cancers, including pediatric sarcomas, neuroblastomas, neuroendocrine cancers, high grade and carcinoid low grades. What we realized or observed in these clinical trials that Seneca Valley virus was very well tolerated. It is to be noted that these previous trials were done with IV administration. So the dose of Seneca Valley virus was considerably higher as compared to what we proposed to do in intralesional injections. And patients tolerated very well, even those high doses. And only one DLT was noted. And even that DLT was tumor site pain, which we understand is because of inflammation from viral infection, T-cell infiltration. And later on, that inflammation would lead to tumor necrosis and pain. One last thing I would like to remind our viewers here that these early trials were done in all comers. TEM8 wasn't discovered at that time. There was no way for us to screen for TEM8. So these were done in all comers. We have since then, the last 10 years, have moved away and have improvised on this. And we now have a biomarker probably the first of its kind for any oncolytic virus therapy to screen patients who would truly benefit from this. Next slide. And with regards to that, Seneca Therapeutics has now developed a pretty accurate assay to bioselect TEM8 expression with the help of 8-gene assay. As you can see here, the prediction of this assay is pretty good with 86% accuracy. Next slide. Now, let's move on to some of the preclinical data, which Paul would be presenting very soon at AACR. And the data is looking so exciting and really revs up my optimism in this concept, especially in neuroendocrine cancers. What you see here is an immunocompetent mouse model because we are testing not only Seneca Valley virus, but also immune checkpoint inhibitors. So what we see here, the mice who got the triplet therapy with Seneca Valley virus and immune checkpoint inhibitor had tremendous disease stabilization, where the animals were alive at 165 days, as compared to all other control groups, where all the animals succumbed to illness at just 97 days. I would also like to highlight the fact at the time of tumor relapse, which is marked by these arrows somewhere around 70 days and about 100 days, we re-injected Seneca Valley virus, and which led to further durable responses and stabilization of the disease. Next slide. We are trying to develop Seneca Valley virus as a true systemic therapy. So the effects are not only just seen in the injected tumors, but we are also wanting to see them in distant metastatic disease. And this experiment here precisely explains that. The lesions which were injected, they certainly were cancer-free several days after the injection, whereas the control group shows tumor progression. However, let me bring your attention to the right-hand side of the screen, the contralateral tumors. Now, these results are very profound. What we notice is that even in the non-injected contralateral tumors, 83% mice were cured of disease, while the other control arms had zero cures. This is pretty significant. And this is also not surprising because we understand how this works. When the tumors which are injected are infected with Seneca Valley virus, this leads to inflammation, inflammatory cascade, activation of PAMP and DAMP, and causing cell death, which releases these tumor neoantigens systemically and really primes immune checkpoint inhibitor to then help them work against other tumors systemically. These Seneca Valley virus also then goes ahead and infects systemically the other tumors which are not directly injected. Next slide. Here, what I would like to bring to your attention is T cell infiltration. As expected, what we are trying to do by injecting Seneca Valley virus directly into the tumor is to get these immune deficient tumors to cause these immune inflammation by attracting T cells there. This is a surrogate biomarker that these tumors would respond to these therapies. And what we see as expected, the mice, the tumors which were injected with Seneca Valley virus and also treated with immune checkpoint inhibitors showed the highest T cell infiltration as compared to any other control group. And I've kind of highlighted that in both the IHC, which is stained as brown, and also quantitative analysis on the bars. Next slide. All right, so that was all preclinical data suggesting some really incredible, promising synergistic effect of combination Seneca Valley virus when added to immune checkpoint inhibitor in immune competent mouse models. And I would also like to highlight the fact that these mouse models also express biomarkers which are seen in neuroendocrine cancers like chromogranin A. However, our optimism is not just restricted to preclinical data. We also have some clinical data which suggest that Seneca Valley virus might be the key to unlock these rare and unmet need cancers. Now, going back 10 years back when this phase 1 study was done, and again, I want to remind the viewers that we had no way to screen patients at that time. TEM8 wasn't in existence. What we noticed that some of the patients in the phase 1 belonged to low-grade NETs or carcinoid tumor category. And we, at that time, knew which patients were able to be infected with Seneca Valley virus based on CXCL expression. Now, retrospectively, when we reviewed these patients, the subgroup of neuroendocrine tumor patients in this pan tumor type phase 1, we noticed those 3 patients who did not develop infection, the median PFS in those 3 was 76 days, as compared to the patients who did develop Seneca Valley virus infection had median PFS of 271 days. This data was presented by Dr. Charlie Rudin from Memorial Sloan Kettering at one of the medical meetings. And this phase 1 study is actually in public domain in a publication. Not only there was disease stabilization in the affected population, what we also noticed anecdotally that some of these NET patients develop hormonal symptoms. You've probably heard of carcinoid syndrome, flushing, wheezing, diarrhea because of excessive serotonin produced by these NET cells. And there was marked resolution of those carcinoid syndrome symptoms in some of these patients who did develop these infections. We now have a very effective way to screen patients who would benefit from Seneca Valley virus, and that is TEM8. Next slide. Now, I'd like to share some anecdotal example from my own personal clinic. This is just a couple of months back. High-grade neuroendocrine cancer patient, progressed on all chemotherapies. You can see how bad the tumor's looking. The patient's literally at the verge of hospice. We've exhausted all treatment options. And then we tried immunotherapy, immune checkpoint inhibitor, and just 2 doses, and the patient's in complete response, back to work, completely normal. Now, we know immune checkpoint inhibitors does have some activity in high-grade neuroendocrine cancers. Doesn't work as well in low-grade NETs because they are immune deficient or considered cold tumors. However, and we were among the first groups to recognize that there could be a role for immune checkpoint inhibitors back in 2018. Next slide. There is some prospective data now. SWOG 1609, it was a multi-center national study, looked into ipilimumab and nivolumab in high-grade neuroendocrine cancer and noticed 44% response rates. So we have optimism that immune checkpoint inhibitor can certainly work in some subgroup of patients and would be the ideal companion to be added to Seneca Valley virus, because Seneca Valley virus by itself has fantastic anti-tumor activity, synergizes with immune checkpoint inhibitor based on our preclinical data. And there is some clinical data to suggest that immune checkpoint inhibitor also has some activity. So this was the premise for us to develop this combination in NET and NEC patient population. Next slide. Now, let me quickly walk you through our proposed clinical trial. The protocol is in very advanced stages. We've had some good negotiations with FDA, and the protocol is currently under review and it's almost ready. We also have endorsement from various NET specialist sites who are eager to activate this study. The phase 1 part of the study, which is the dose finding phase, has 6 distinct cohorts. The first 3 cohorts, we would be testing single injection of Seneca Valley virus in addition to immune checkpoint inhibitor at increasing doses. And the next 3 cohorts are going to test multiple dosing strategies of Seneca Valley virus in combination with immune checkpoint inhibitors. We also have a dedicated cohort where we would be doing pre-treatment as well as post-treatment biopsies to really study the effects of Seneca Valley virus, the viral replication, and various other exploratory correlative studies. Next slide. Once we have completed the phase 1 dose escalation study and we have established a recommended phase 2 dose, the study would then rapidly move into a more definitive randomized control trial, a phase 2 study. And this is all done under the umbrella of the same protocol. So there would not be any lags. Our 2 disease types that we are interested in this randomized phase 2 are neuroendocrine cancers, the high-grade aggressive cancers, as well as low-grade well-defined NET. Patients would be randomized in 2:1 fashion in favor of Seneca Valley virus, ipilimumab, nivolumab combination versus just ipilimumab, nivolumab. And we would be looking for primary endpoint as median PFS. Next slide. Based on some of the historical data with immune checkpoint inhibitors in NEC and NET, we believe that by addition of Seneca Valley virus, we should be able to see at least 75% improvement in the median PFS, just as compared to immune checkpoint inhibitor by itself. And in the NET cohort, which are traditionally not very immunogenic, we believe that about 46% improvement. So as you can see, we are quite optimistic based on the preclinical data, the clinical data, as well as the historical data. We believe that we can certainly improve on the current standards. I would also like to bring to your notice that just 18 patient small SWOG 1609 study, SWOG DART 1609, led to NCCN recommendation of ipilimumab in NEC group, or poorly differentiated neuroendocrine carcinoma, which I heavily use in clinic. So that's how low the bar is because of the critical unmet need in this disease type. Next slide. The primary objective of phase 1, as I mentioned, is to look for the safety profile of the combination as well as the recommended phase 2 dose. And the phase 2 is designed a randomized controlled phase 2 study, first of its kind, with using oncolytic virus and neuroendocrine cancers, and is looking for median PFS differences. Next slide. And not only these efficacy endpoints, we are also woven in a lot of interesting science into this phase 1 and randomized phase 2 study. We will be studying, next slide please, we would be studying viral replication, we would be studying impact of Seneca Valley virus on various cytokines. We would like to look for biomarkers of response as well as non-responders. So this study is truly very unique, and we have a lot of provisions for collection of solid tumor tissue as well as liquid tissue and stool tissue in these patients to study them further. With that, I'd like to conclude my talk, and over to you, Paul. Thank you.